Welcome back, friends. We are talking the first video we talked about what is apoptosis or programmed cell death and why apoptosis is so important. Now we have seen two different pathways of apoptosis, intrinsic pathway and extrinsic pathway. Okay, and we've seen the difference between these two pathways, but we also know that actually both of this pathway lead to the destruction of the cell and killing of the cell. That is a function of programmed cell death. That means the cell, when it goes through any kind of stress or any sort of problems like DNA damage and stuff, it try to kill itself to minimize that effect to only that cell, not to the neighboring cell. So these are the ideas, and we know intrinsic pathway generates from inside the cell, extrinsic pathways on the other hand generates outside. The pathway source generates from the outside. Now in this video tutorial we'll be talking about the intrinsic pathway of apoptosis, or we also known as mitochondrial pathway of apoptosis. So this is the intrinsic apoptotic pathway or mitochondrial pathway. So let's talk about the intrinsic pathway in much more details. Okay, intrinsic pathway of apoptosis. This is also known as apoptosome mediated apoptosis. Why? We are going to know in a moment. Now intrinsic pathway, why it's called intrinsic? Because the origination of this pathway is inside the cell. So if I draw a cell here to explain it in much better quality, then this is the cell, and normally this intrinsic pathway originates inside the cytosol, and it affects mainly start point of this process is the mitochondria. The target is mitochondria, and for this process to activate, they have different types of problems in cell. Problems like endoplasmic reticulum stress or any sorts of stress going to the cell. Problems could be a DNA damage. So whatever type of response, we can get that response from outside or from inside. In this case, most of these responses we're getting from inside. For example, the DNA damage. DNA damage. DNA damage is a signal that a cell can get from inside. So if the DNA is broken down in some fact, in that case, that acts as a signal. And we know that there are certain signaling molecules that are found inside the cell who sense this DNA damage and stuff. And some of those examples are proteins like ATM, proteins like CHK. These are the proteins. So once these are the sensory proteins, once they sense the DNA damage, they activate P53. P53 once activated, it is a very dangerous protein because P53 can turn on multiple proteins and other mediators who will ultimately guide the cell through apoptosis. Now I also told this thing in case of how cell cycle is regulated, but in this case, you know, once the cell is, let's say the cells start to divide, and that time it's found out that DNA is broken, so P53 is activated. Now P53 will not allow this cell to pass to the next stage, or phase of the cell cycle. It will halt the cell cycle for this cell. So now the P53 will start recruiting other factors or other proteins there, P21 and many more other proteins. Such some of the protein, one of such protein is BAX. BAX is a protein that can create pores in mitochondria. So if I draw mitochondria, let's say this is a mitochondrial drawing. Let's say this is the mitochondria, and BAX creates pore in the mitochondria. BAX proteins creates pore. So it creates pore in the middle. So as BAX creating pore in the mitochondria, it will allow a very important component, cytochrome C, to be released from mitochondria to the cytosol. Okay, so what it does, it helps in releasing cytochrome C. Once cytochrome C is released in the cytosol, this is acting as a death signal, because this should not happen. Cytochrome should be present inside mitochondria. Once it's coming out to the cytosol, the cytochrome C will pair with another protein called APAF, a protein called APAF. APAF1. So cytochrome C and APAF1 together will catalyze a very important activity, and they will activate a set of protein cascade reactions in the downstream in future, and that cascade of reactions will ultimately turn into a nuclease activation and protease activation events. So this cytochrome C and APAF1 will go and activate caspase 9. Previously the caspase is present as procaspase. Pro means early, when it is not active, it's inactive. So this APAF1 and cytochrome C together is going to activate procaspase into caspase, right? So now caspase 9 means this caspase is active. So once caspase 9 is active, it will go and it will further activate procaspase 3 into caspase 3. So now procaspase 3 become caspase 3. Once caspase 3 is active, caspase 3 once active, it is going to further activate nuclease enzymes. How? Normally nuclease enzymes are inhibited by another sort of molecules. Okay, nuclease is inhibited by that molecule, but what caspase 3 does is that caspase 3 will go, let's say this is the nuclease enzyme, and this enzyme's activity is blocked by this inhibitor. Okay, now this caspase 3 will go and it will degrade this inhibitor. It will degrade the inhibitor. So as it degrades the inhibitor, it remains the nucleus active because inhibitor is now taken away. So this nuclease becomes active. So once nuclease become active, nuclease enzyme can go inside nucleus and can find DNA, can find the chromosome, start degrading them. So once they start degrading the DNA inside nucleus, the cell cannot survive. So as the protein synthesis is halted, proteins are also degraded, and also this caspase 3 can also activate some proteases also. So, and P53 in the other hand, this is a Bax signaling pathway, but on the other hand it can also activate some proteases. They will break down proteins. So ultimately protein breakdown, nucleic acid breakdown leads to the generation of the fragmentation of cell. So they start forming bulges because microtubule is getting distorted, actin filaments, lamins get distorted. So they the nuclear structure gets distorted because the nuclear lamins are degraded. So as a result, they start forming the bulbs. We call them the blebs, the apoptotic bleb. So once those things formed, then the cells are destroyed, then the macrophage will come or any phagocytic cell will come and engulf those portions. This is a general way of how intrinsic pathway works using mitochondria. Now there are multiple details. You probably seen books, many more protein names and things, they are working together and stuff, but majorly this is how this whole thing works. Okay, now one thing I must tell you regarding this point of Bax signaling, because if P53 is active, Bax will be active. So this pathway of Bax activation, and Bax, what you will do, Bax is going to cleave and pour holes in the mitochondria. So to prevent that, we have another protein there to prevent Bax, and that is BCL. So BCL blocks the activity of Bax, because you know in biology we should have a negative regulation also, otherwise it's going to hamper the process all the time. So we have a protein called BCL2 which is going to regulate this purpose. BCL2 is going to halt or inhibit the activity of Bax, right? So if BCL2 is active, then Bax will not function, but upon activation of P53, P53 on the other hand inhibits BCL2. Okay, so P53 in turn inactivates BCL2. That means P53 is helping Bax and also inhibiting BCL2 so that Bax can function properly. So this is a regulation feedback that is present that you should know. Otherwise there are multiple things, but for understanding an overview of how intrinsic pathway works, This is how it works. Okay, so I hope this video helps you. If you like this video, please hit the like button. Here's the subscribe button. Hit the subscribe button to subscribe, and what subscription will give you will send you a lot of videos like this which will be informative and important for you. Thank you.